Good afternoon, everyone. On behalf of CIRM, the scientific programs, the review, and the grants management teams, I would like to welcome everyone to this informational webinar that will focus on our DISC2 Quest Program Awards. This is now the the fifth of our webinar series that we have created as resources for applicants interested to apply to CIRM funding. The information contained in these webinar presentations and the Q&A will be made available through our website along the FAQs derived from our interactions with all of you. We hope that this will become a resource for for current and for future applicants and help answer many of the questions that you encounter. The goal is that applicants to the DISC2 program consult with CIRM staff only when there is a need to discuss the specifics on the research project and how does it fit into the program. However, the rest of the information should be able to be found in these webinars and the resources on our page. We're excited as today's webinar brings a novel and very needed path to this program that we will be describing shortly. Next. As always, this program is a part of our mission, which is to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and the world. Next. For familiarity, the DISC2 program has been developed and is scientifically managed through the scientific programs and education team. You can see here the members of this team. We have Dr. Kelly Shepard, who is our associate director, and she's been working together with Dr. Chanlek Tang in the updating of this program. But we also have many of our other colleagues, Dr. Ukta Grisamer, Dr. Janie Byron, Dr. Daisy Chin, and Dr. Sarah Taylor, who together with Kelly and Chan, they contribute to many of the calls for consultations and they manage many of the awards. Next slide. During today's webinar, we are actually also honored to have two of our colleagues, as well as Kelly and Chan, who will be helping us with many of the sections and the questions. The colleagues are Dr. Haley Lam, who is Associate Director of Portfolio Development and Review, and she's in charge of all the parts that have to do with the review of this program. And April Reinhardt, who is the Grants Management Officer that helps us manage all these grants and that can help us also answer many of the questions about this program. Next slide. DISC2 is part of our pillar program offerings. Next. This program is the first step into our CIRM's R&D pipeline, and the scope of the DISC2 Quest Awards corresponds to early translational research with the ultimate goal of leading to a single development therapeutic or biomarker candidate, which is what's new this time, ready for further development into our translational or clinical programs. Next. This slide provides a high-level overview of all the discovery program opportunities, and you can find the details in our programs page for the DISC2 and the Remind-L, also known as DISC4, and the upcoming Remind-X, which we don't have details yet. Next. What we are excited about today is to introduce the novel path that we have created within the Quest Award program. And besides the original therapeutic candidate path, for the first time, CIRM has created a specific program track dedicated for research supporting identification and development of biomarker candidates. Our team will be going over the details of both these programs in the upcoming slides, so I'm not going to dwell into any more of the in this slide. With that said, we've organized today's webinar in two parts. Next slide. The first one is a presentation that will go over the overview and updates on those two tracks, the application process. Dr. Haley Lam will go over the overview and resources and context, as well as hopefully we have time for common issues. And we would like to also have time for questions, Q&A. afterwards. So without further ado, I will leave to Dr. Kelly Shepard to go over the first part of our program overview. Thank you.
Thank you, Dr. Canes-Aviles. So I'm going to be going over the DISC2 program by starting with a general overview. Next slide, please. Now, Rosa already just put the Quest Awards in the context of our other discovery programs for you, but I'm just going to remind you that this is the first step on a path that is intended to ultimately culminate in a development candidate that will be translated into clinical use and eventually go to a clinical trial. So this is the first step along this pathway. And the goal of the DISC2 award is to identify a single product candidate that can immediately move into the stage of study supported by our translation stage program. Next slide, please. So regardless, could we go back one, please? Yes, so regardless of whether we are pursuing a therapeutic or a biomarker path, all DISC2 programs must propose a type of development candidate that is deemed eligible under the program announcement and must also propose an experimental design that is put in place in order to achieve the expected outcome. And I'm going to go over both eligible candidate types and expected outcomes in my following slides. Next slide, please. So these are the two tracks, the therapeutic track, which is not terribly dissimilar to the traditional DISC2 therapeutic candidate path that has been going on since 2016 now. And then the new biomarker track, which we will go over in a little bit more detail subsequently with my colleague, Dr. Chan-Lek Tan. But what's different this year compared to previous rounds from therapeutics is that we've increased the budget to $1.75 million in direct project costs. Other elements are not too different as far as the therapeutic track goes. It's open to California nonprofit or for-profit research institutions. There's a single principal investigator who must commit a minimum of 20% effort. And a successful DISC2 program will culminate in a development candidate that will be ready to compete for funding through one of our translational stage programs, ideally. Next slide, please. Okay, so what are the eligible candidate types for therapeutics? These can be grouped into kind of four major categories. The first category is a cell therapy candidate. So this would be a population of cells that is either human stem cell or progenitor cells that compose the therapeutic candidate, or it could be that it's a cell therapeutic that is derived from human stem cells or progenitor cells. In other words, a differentiated derivative, for example, pluripotent stem cell derived islet progenitors or something of that sort. The second category is a genetic therapy. This is defined in the CIRM program announcement as a therapeutic intervention that alters genomic sequence or introduces or directly manipulates nucleic acids in cells and targets a human somatic cell for its therapeutic effect. And key to being relevant to CIRM's mission is intended to replace, regenerate, or repair the function of aged, diseased, damaged, or defective cells, tissues, and or organs. The third category here listed under C are stem cell relevant projects that are not made of stem cells themselves. This could include things like small molecules or biologics, peptides, antibodies, etc., that act on endogenous human stem cells for their therapeutic effect, or it could be an entity that targets a human cancer stem cell for its therapeutic effect, or it could be an entity that modifies a stem cell therapy, or finally, one more example is it's an entity that requires human stem cell for its manufacture. So an example of that could be an exosome product that is derived from a stem cell. Now, important for this type of approach is you should be prepared in your application to provide justification that it is indeed acting through a stem cell mechanism, whether it's an endogenous stem cell or a human cancer stem cell. You need to explain how the stem cell relates to the therapeutic that you're developing. Lastly, other modality that uniquely requires human stem cells for candidate identification and testing. An example of this would be a small molecule drug that acts on a neuron, where an iPSC-derived neuron screen, a screen using iPSC-derived organoids or neurons, was used to identify that candidate. So the candidate itself is a small molecule. It doesn't really have anything to do with stem cells in terms of its mechanism of action. However, those stem cells were uniquely required in order to identify and test that candidate. So for this type of a program, you also need to prepare to justify the requirement for human stem cells in your approach. Next slide, please. So those are the eligible candidate types, but I also told you earlier that in order for a Quest application to be eligible, you must also propose a research plan that is designed in order to achieve the expected outcome. So in the case of a therapeutic, the expected outcome is a candidate that is ready for translation. And what do we mean by ready for translation? That means by the end of your DISC2 award, you will have selected a single therapeutic candidate of one of the modalities I described above. You will have developed a draft target product profile to accompany that therapeutic candidate. You will have developed some preliminary measures of identity, activity, and purity. And most importantly, you will have demonstrated reproducible disease or injury modifying activity with that candidate. Now, all therapeutic candidate proposals should aim to achieve this expected outcome. This is the bar that you need to meet in order to be ready to apply for CIRM's translational stage programs. And if you look at a TRAN program announcement, you'll see that the entry requirements are mirrored very closely to these exit requirements for our DISC2 program. Now, I'm going to go into a little bit more detail about the disease modifying activity because that's a really important part of a Quest Award proposal. Next slide, please. So in order to demonstrate disease modifying activity, there are a few other things you should be aware of as you're putting together your research plan. If you are proposing to develop a cell therapy that is allogeneic, that is going to be coming from a donor source or cell line, you must design your experiments to culminate in a demonstration of disease-modifying activity or efficacy using the therapeutic product that was made from a clinically compatible cell line. What does that mean? That means the cell line meets the good tissue practice requirements for donor eligibility that the FDA prescribes and has been appropriately consented for intended use and for clinical development and sale. Now, if you're working with pluripotent stem cells in your lab and developing a therapeutic, you need to make sure that you have a line that meets these requirements to use and ultimately be the basis of your candidate in your Quest Award proposal. If you don't have a line or if you don't know if your line meets these requirements, we'll provide a little bit more information about that for you later in this webinar, but there's also more there's also more information linked in the appendix of our program announcement. Now, if you're developing an autologous cell therapy, where the cells are going to eventually come from the patient that would be treated by this candidate, disease-modifying activity must be demonstrated with candidates derived from 2 or more donors to establish reproducibility. So you should design your research plan to culminate in the efficacy study with a product made from at least 2, possibly more donors. If you are proposing a genetic therapy candidate, you will need to provide evidence demonstrating that the selected candidate targets or has activity in a clinically relevant human cell population. So you may be demonstrating disease-modifying activity using animal models or in vitro models or in human pluripotent stem cell-derived models. You will be proposing and justifying the models that you choose to use. But at minimum, you must also have evidence demonstrated that your final gene therapy candidate can target or has activity in a clinically relevant human cell population. Finally, for those other modalities, small molecules, biologics that target endogenous stem cells or cancer stem cells, similarly to the genetic therapy, you must demonstrate disease-modifying activity on or with a relevant human stem progenitor or cancer stem cell population, in addition to whatever animal models you choose to use. Next slide, please. So I've just gone over the expected outcomes and the eligible candidate types. This is a list of the different types of activities that are pretty typical of a Quest Award application that support achieving those outcomes. So studies to identify, test, characterize your candidate. These are the types of assessments that can eventually form critical quality attributes or assessments of safety or biodistribution necessary for candidate selection. We're not talking about the pivotal safety studies that are done later once you've already demonstrated efficacy. We're talking about assessments of safety or biodistribution necessary for you to choose between different, perhaps, variations of your candidate to decide which one is the best to move forward with. Testing therapeutic feasibility, reproducibility assessment. Optimization of candidate. This can include testing different cell lines, testing different variations on your vectors, enhancers, promoters, etc. If you're working with small molecules, developing and identifying structure-function relationships, your proof of concept studies and demonstration of disease-modifying activity, developing the target product profile, and preparation for and conduct of appropriate regulatory meetings. An appropriate regulatory meeting for this stage of research, DISC2, is most likely going to be your INTERACT meeting with the FDA. Okay, next slide, please. All right, with that, I will hand things over to my colleague, Dr. Chan-Lek Tan, who will tell you a little bit more about the biomarker track. Thank you.
Great, thanks, Kelly. As Rosa and Kelly mentioned earlier, the DISC2 program has been expanded to support biomarker discovery for the first time this year. So we will spend a little bit more time on this section to explain this new track. So what are the eligible biomarker candidates? The first category of biomarkers are those that support therapeutic development or clinical use of the following therapeutic types as described earlier by Kelly. So cell therapy, genetic therapy, or other modalities that act on or target human stem cells or cancer stem cells, modify stem cell therapies, or require stem cells for manufacture. So I won't repeat all the same requirements hold true here. With strong justification, this award will also support biomarker discovery for other modalities that are not covered here, but for which stem or progenitor cells are uniquely enabling. So for example, if human stem cells are an integral part of the biomarker assay, we strongly encourage you to reach out to the discovery team, program officers, if you plan to submit an application under this latter category. We should emphasize here that superficial or trivial use of stem cell modeling for testing purposes will not be deemed eligible. All right, and actually before we move on, I just want to kind of clarify some things within these requirements that are laid out here. All biomarker categories as defined by the FDA BEST glossary are eligible. So response biomarkers, predictive biomarkers, diagnostic biomarkers are all eligible, as long as you can make the case that it belongs to one or other of these categories here. Furthermore, your proposed biomarker can be of any modality or based on any technology. So they can be molecular, histological, imaging, and so on. All right, next slide. Regardless of the type of biomarker candidate being proposed, and similar to the therapeutic track that Kelly had outlined, you must propose studies that can lead you to the following expected outcomes by the end of your award period. So these expected outcomes are selection of a single biomarker candidate and specification of its context of use, development of a draft biomarker candidate profile. I can talk about this a little bit more, but it's modeled on the letter of intent submission template that is developed by the Biomarker Qualification Plan by the FDA. Selection of an analytical method, demonstration that biomarker can be reproducibly measured at relevant levels to your context of use, and initial proof of concept using relevant clinical data or samples. Next slide, please. This slide helps to illustrate some of the types of activities we expect to support, and it's not meant to be exhaustive. So some of these activities were already previously covered, but it's helpful to highlight a few of them here. So in your proposal, you may include unbiased screening or profiling studies, as well as studies that seek to clarify the relationship between your biomarker and the biological processes underlying the context of use. You may also undertake studies that are meant to refine or modify your proposed context of use. You may also test or compare measurement methods or develop custom assays to measure your biomarker. And we will also fund the development of algorithms, indices, or other necessary data transformations for the use of your biomarker. We've already touched on the proof of concept. That is one of the expected outcomes. We also will support preparation and completion of appropriate regulatory meeting or submissions. Just a quick note here, some of the requirements and the materials in this program has been in part informed by the FDA biomarker qualification process. And we have included links to the BQP website, and we encourage you to use these resources and determine if it's relevant to your program. That being said, the DISC2 award has no requirements on the regulatory pathway for your biomarker. So you're free to seek regulatory approval through the most appropriate pathway for your program. And we encourage you to explore all the opportunities for exploring FDA advice where appropriate. And so these are the activities that are also allowable under both tracks. So activities intended to promote and uphold principles of DEI in the conduct of the study, as well as activities that are associated with data sharing and knowledge sharing from the study. Next slide. And some of these activities are not generally supported under the DISC2 program because they're sort of further down the development pipeline. So for example, activities that are covered by the TRAN program, including development of GMP and clear compliant process, filing of IND or 510K, or planning for commercialization or conduct of clinical trials would generally not be supported under the DISC2. And for the biomarker program, we will also not support clinical final clinical validation studies for your candidate biomarker that's necessary for final regulatory acceptance. All right, and I'll hand it back to Kelly to talk you through the application process.
Thank you, Chan. Next slide, please, April. Okay, so this might be familiar to those of you who have applied for a CIRM award before, but if you haven't, the first step in starting an application is to visit the CIRM grants portal, which is at that website you see there, grants.cirm.ca.gov. If you're new to this, you'll set up a login and password. If you're not new to this, you will log in using your existing login and password, and you will visit the link marked Open Programs shown there in the box. There you will see a list of RFAs and programs that are open for applications, and you'll click Start a DISC2 application as you see in the orange box on the lower half of your screen. Next slide, please. Once you open an application, you're going to see a menu on the left side of your screen that lists all of the individual components of the application and their status. As you go through these and complete them, they will go from incomplete to complete. Now, a good portion of these sections that you see are completed online, including eligibility, the places where you enter the key personnel, information, some basic information about the project, including the positive selection preview page, which we're going to come back to in a few minutes. Sections where you describe the budget, including any subcontracts that would be a part of this award, and your certifications for any institutional oversight or assurances that you might need or be required for your project. Down towards the bottom of that menu, you'll see a section called Uploads. This is where you will have some templates that you can download that are required parts of the application. You will complete them offline, and then you will attach them to this online application at the time of submission. Back up, please. Just briefly, the required templates, and we're going to go over these in a minute, include the proposal itself, the biographical sketches, the PI other support, and for-profit applications, there are a couple of special uploads that you'll see that we'll go over in my next couple of slides. There are also optional slots for uploads that include, that is there if you have letters of support, quotes, or material transfer agreements, or other documents that could be useful for the application. Next slide, please. Thank you. So I just want to back up for a second and talk about the positive selection preview page. So the reason we want to call this to your attention is because it's a very important part of the application that is not often or not always recognized as such by first-time applicants. So this is going to appear as sort of a high-level executive summary where you're prompted to answer a series of questions about your project. This may appear duplicative with what you're going to be writing in the uploaded proposal. However, this is an executive summary, and it's the basis of which our two-part application process begins. So Haley will be describing the review process later, but it's a two-stage process, and this is the first place that reviewers are going to be looking to begin the process of review. And we will go over that again, as I said, in the next couple of slides. So please pay attention and devote significant attention in the completion of this page when you're working on your application. Next slide, please. Okay, so I wanted to highlight the budget section because there have been some changes to this made since the last time some of you might have applied for a DISC2 or any other discovery stage award for that matter. So normally there has been a table up near the top of the funds calculator which is where you enter the request by year for the direct project costs, etc. So previously it was just a table with direct projects costs, but now it has been broken down into several line items, including supplies, equipment here. So we just want to make sure you're aware of that and you allocate the right amount of time to put together the budget if you hadn't, if this is something that you perhaps weren't expecting. Now, going along with the CIRM funds calculator is the budget justification narrative, which is right below it in the left there. All costs should be that are listed in this direct projects cost table by the categories by the year should be justified in that narrative in sufficient detail that you can that the amounts listed in the table make sense to the reviewers and to CIRM administrators if this award is eventually funded. Next slide, please. Okay, here's a close-up look at the upload section. So this is where you will be finding templates that you need to use to complete parts of your application. So for example, at the top there, you'll see the proposal template. That's a Microsoft Word document. You can click on that link there to download it, complete it offline on your own time. That's where you'll have your figures, your research plan, your preliminary data, your references, a lot of the meat of your proposal. Once that is complete, you will convert it to a PDF, and then you will upload it there where it says on the right, choose file. These are all the ones that say status incomplete are the required documents, and you must use these templates that are provided in this table for you and when you put together your application. Now, this is an example screenshot from a not-for-profit application. If you are a for-profit applicant, you will notice a couple of additional required uploads listed in this table when you open your application. Next slide, please. So again, here are the required uploads and what the templates look for them. Over on the left, the proposal, we have the biosketch in the middle and the other support. I might sound redundant, but you must use the templates found in the grant application. CIRM will not accept templates from other institutions or funding agencies. Okay, next slide, please. These are the two uploads that are required for applicants only. Over on the left is the financial feasibility and assessment and solvency form, which is a Word document with a checklist on it. And over on the right is the financial solvency template, which is an Excel spreadsheet. Again, you must use these provided templates found in the upload section. Next slide, please. So again, with respect to the required uploads, do use the templates that are provided for you and do adhere to the prescribed page limits, font sizes, formatting that are already programmed into those templates. Follow the instructions carefully for each section, and when you're done, convert these to PDFs to upload. Do not alter the margins or exceed the page limits. Don't leave any sections incomplete, and as I mentioned previously, do not use a divided. Next slide, please. And the last point I want to touch on, some of you may have submitted applications in past cycles of DISC2 and are resubmitting. If you were scored in a previous cycle and will be addressing reviewer feedback, there's an area in the information for review section online where you can check a box that indicates that you are submitting a substantially similar proposal that addresses Grants Working Group reviewer comments. There's a place for you to enter the application number. And in the proposal template, you will find a page called the resubmission statement where you're allowed to go point by point and address those previous critiques. If you applied in a previous DISC2 cycle and did not pass the first stage of a review, positive selection, you won't actually have any score or feedback to address. And so you would just submit as a new application. Next slide, please. Okay, I'm going to now hand things over to my colleague, Dr. Haley Lamb from the review office, who will give you a few more details about the review process.
Thanks, Kelly. I'll be just going over a couple of process things for what happens once your application is submitted. Next slide. So after you go through all the things that Kelly just went over, your application will finally be submitted to CIRM. And then it comes to the review team. And there's 3 major steps in what happens to your application up until hopeful funding of that application. So the first part is that we do a quick check of every application just to make sure that all of the pieces have been submitted that need to be submitted, and you haven't done things like go over the page limits that Kelly mentioned in the proposal and things like that. So all of that are checked over internally in CIRM. And once it goes through all of those things, and we also, the other thing that I should mention is that we do check for the eligibility scientifically as well. So all of the guidelines that have been discussed in terms of the types of projects that are eligible for both the therapeutic and biomarker tracks are things that are verified at this stage as well. So once the applications go through that, they get accepted formally for scientific and merit review. And that's where we bring in together a panel of 15 scientific experts, as well as up to 7 members of our board who review all of the applications. In the first stage, there's the positive selection stage. And so all of the applications that move forward from the eligibility stage go into positive selection. And I'll be discussing that a little bit more in the next slide. And then it moves applications that have been, oh, wait, not yet. Yeah, so then the applications that move through to the final GWG discussion will be evaluated individually by the panel and discussed, where there will be a final GWG score and recommendation. Those recommendations and scores go to the board for final funding decisions. All right, next slide. So a little bit about the positive selection. So this is pretty standard practice for our discovery programs, where we tend to get a lot more applications than can be reviewed in full. And so all of the grants working group members are allowed to select up to a certain number of applications. And again, this is the group that includes the 15 scientific experts, as well as the patient advocate and nurse members of our board who sit on our grants working group panels. And they can conduct a pre-review of those applications and select which ones move forward to the stage 2 full review. And again, these applications can be selected by any member of the grants working group. And after that, the CIRM president and scientific team can also take a look at any applications that haven't been selected and decide if they want to move them to the second stage. So important note that any applications that are not selected and that do not move forward to stage 2 do not receive any official critiques or feedback. And so I understand that this can be really tricky, but essentially we don't have any formal feedback for any applications that are not selected. All right, so second, the next stage, please. So the next slide. If an application is selected, it moves forward to the full scientific review. And for this, all of the applications that are up for discussion are scored on a scale of 1 to 100. So a score of 85 to 100 is a recommendation for funding to the board. And a score of 1 to 84 is a do not recommend for funding. And a note here that if your application receives a score of 80 to 84, so basically just below the recommendation for funding range, that application can be resubmitted in a future DISC round and can bypass that positive selection step that I just described. That being said, all applications that receive any score can always revise and resubmit in a future round. Next slide. And then just a note about the review criteria. The holistic scoring of 1 to 100 is across these 5 criteria. And this has not changed from prior rounds and is outlined in more detail as well in the program announcement. And with that, I think I'll pass it back to the scientific programs team. Thank you.
Great. Thanks, Haley. So as we close out the webinar, we just wanted to highlight some useful resources for applicants on our website. Next slide, please. So on our website, you'll find our guidance for data sharing and management. CIRM requires that all the data generated using CIRM funds are shared using FAIR principles. And you will find on this page our templates for data sharing and management plan, which will be required of the awards that are funded. And so you'll see our expectations for funded applications in this regard here. We also have guidance and information on clinically compatible cell lines. As Kelly mentioned earlier, these are particularly important for applicants seeking to develop allogeneic therapeutics. So you'll find the information around what constitutes clinically compatible cell lines, as well as there's a link here that we've shared this useful informational webinar that was conducted on the topic led by Dr. Ross Okamura in 2022, which you should check out if you're unfamiliar with the topic. On this page, we also have a list of partners through our CIRM Industry Resource Partner Program, where partner organizations have worked with CIRM to provide CIRM researchers with access to their clinically compatible HPSC lines under a standard agreement. So that's a resource you can find here as well. Next page. Another useful resource on our discovery page is these review summaries from previous rounds of DISC2 awards, which is very helpful for you to get a sense of the types of proposals that have been successful in the past and the kinds of comments that have been made by reviewers in their panel discussion. And next slide. So for general programmatic questions, we encourage you to reach out to the discovery team at discovery@cirm.ca.gov if your questions are not already answered by this webinar, and we will schedule a Zoom consultation to discuss program fit if we deem it necessary. If you have specific questions on budgets, administrative and review processes, you can also reach out to our respective teams here at these addresses. As we mentioned, this webinar recording, the slide deck, and the FAQ document will be posted to this webpage in about a week from today. And a final reminder that the applications will close on May 14 at exactly 2 p.m. Pacific time. All right. So for the next slide, I think we have some time. So before we go on into the Q&A, we will take a quick second to highlight a few of the common issues that we have encountered over the years. Next slide. These are broadly in the categories listed here. They are either in an ineligible candidate type, proposal do not achieve the expected outcomes. You have maybe proposed inappropriate activities that are out of scope for this award, or there are serious application submission issues. And I can briefly go over some of the examples here. So next slide. So for ineligible candidate types, what we see frequently are candidates that are non-stem cell or non-genetic therapeutics. So in this category, you could have a modality like a small molecule or an antibody that is proposed to act on a stem or progenitor cell, so category C, but the target population is not commonly understood to be a stem or progenitor cell, so you will get some questions to clarify this. Another example is where you have proposed a therapeutic that is not a stem cell or genetic therapy, but has a proposed unique requirement for stem cells for candidate identification and testing, but the application fails to adequately support this contention. So as I mentioned earlier, trivial or sort of superficial use of stem cell modeling for testing will not be deemed eligible. Next slide. The second basket is around applications not including studies that will lead you to the expected outcomes that we seek to have by the end of your award period. So these quotes from these kinds of proposals could include something like, after selecting the candidate, we plan to demonstrate disease-modifying activity of our candidate in a future award. And that's just clearly not what we're looking for. You will have to, again, to reiterate, you will have to both select your development candidate by the end of the award and demonstrate disease-modifying activity of your therapeutic in a relevant preclinical model. Next slide. So out of scope activities include, plans to conduct a clinical trial or further process development, including GMP production and scale up of manufacturing without first establishing that you have disease modifying activity. And these are beyond the scope of a DISC2 award. And then finally, serious application submission issues include not using the templates or grossly incomplete applications. With that, I think we'll hand it back to Sarah, who is going to moderate our Q&A.
Hi, thanks, Chan. So we'll jump into the Q&A now. I'll start with a question that we've seen quite a few times here and with registration. So the first question, are there any focus areas or priority areas for this round of DISC2?
Are you going to call on us or should we?
Either, I guess, Kelly, you can take it.
Okay. In this program announcement, there are no, CIRM is agnostic as long as the modalities are of the types that have been defined as eligible. It is up to you to make the case for the significance and impact and the feasibility, et cetera, the items that are part of the review criteria. So there's no priority given to certain areas by CIRM or CIRM reviewers. It's really about the significance and the impact and the things that you will describe in the proposal and articulate as the value proposition.
Thank you, Kelly. Let me just add something here. This question is probably because we had added the neuro priority before. As everybody remembers, there are $1.5 billion allocated to diseases of the brain. And we have a neuro task force that has led to the development of the Remind DISC4 concept. We have also Remind X coming up. So the neuro is kind of taken care from there. We are not prioritizing that specifically. And there's also strategic allocation framework happening right now. And in September, in future calls, things might change. But for now, as Kelly said, no.
Okay, great. So next question is, we have had quite a number of questions. So this one will go to April regarding new companies that do not yet have funding and asking whether they need to complete a solvency template and other requirements for things such as California eligibility if they do not have employees yet.
Thank you. Yes, it would still be required because you have to still demonstrate the 180 days cash on hand and contingency funding. And I'm sorry, what was the other part of the question, Sarah?
Yes, whether California organizations, if they don't have employees yet.
Yes, you do have to have at least one employee that meets CIRM's definition of a California organization.
And then here's another eligibility question in terms of California-based company is whether an individual can serve as a PI if they work remotely for a California-based company. I think this is for an out-of-state PI working for a California-based company. So I think this one can go to April again.
Thanks. I'm going to share CIRM's definition of a California organization, and that might cover several of our questions, if that's okay. So a California organization by CIRM's definition is a for-profit or nonprofit organization that employs and pays more than 50% of its W-2 employees who are paid and are required to file California state income taxes in California, directs and controls the award activities from California, and retains exclusive rights to intellectual property arising out of the CIRM-funded project. And it's still one minimum required person to meet that criteria.
I can add that a principal investigator can be contracted by the California organization, and you could even technically contract somebody out of state to act as the PI. However, and April, please confirm that if I say anything incorrect, the 20% effort requirement of that PI has to be expended in California.
Correct.
And then we also have several questions about eligible projects. So this would be things like engineered T or NK cells, and epigenetic editing. Is epigenetic editing eligible? Chan, would you like to take this one?
Yeah, I think we can't say anything that's generic here, but for epigenetic editing, I think we would consider that in the spirit of genetic therapies. Obviously, it's a little on the edge there, but you should get in touch with us to have a deeper conversation about that.
Yeah, and then that applies for general eligibility questions about specific project questions. We have another question asking if we have two applications, each meeting a different candidate type category, so one for cell therapy and one for small molecule, can we submit two different applications? Haley, would you like to take that one?
Yeah, so technically, yes, but it cannot be the same PI. So there is a limitation that for any round. So for example, for this application cycle for DISC2, it is one application per PI. And I would just say also to just something to be careful about with those applications is that we do look for overlapping activities. So if, just don't have overlapping activities across multiple applications.
Okay, and then another question we have is, are there restrictions on eligibility of applying PIs? So are instructors eligible to apply, or does one need to hold an assistant professor or higher appointment? Kelly, would you like to take that one?
Sure. So the PI is the person who's going to be accountable for the conduct of the entire research project and the reporting to CIRM. And so it is really up to the institution who they're willing to stand up as a person that can take that role. The PI is really important as well from a reviewer's perspective because their track record of success and their expertise and perhaps their leadership will be something that reviewers might consider when determining the feasibility of a project. So the short answer is if your institution is willing to stand you up as a principal investigator, an instructor could technically be the principal investigator.
Okay, thanks. And then the next question is, how much preliminary data is needed to apply for DISC2?
I can take that one. I think we don't have any formal stipulations on that for DISC2. You should consider whether you are likely to meet your expected outcomes within the award duration that were laid out earlier. So if you're a project that is very, very early, I think it's quite unlikely that you will meet those expected outcomes. But if you have confidence that you can do that, then we have no restrictions against that. But just to bear in mind that this is a pretty competitive award. So the projects that come in with some degree of significant preliminary data would naturally do better.
Okay, great. And then we've also had a few questions asking whether there can be collaborations of multiple PIs for DISC2.
I can answer that. So the CIRM application will only recognize one individual as a PI who's ultimately accountable. However, key personnel, which are other members of the team, and collaborators, which are other members of the team, can have their role defined in the application as to what their contribution would be. So while they wouldn't be seen as a co-PI in the sense of managing their own separate budget, it would still flow through the main principal investigator. You can certainly indicate by your descriptions of key personnel and the percent effort that you're and the support provided to your collaborators or key personnel, their contributions and how their expertise is important to the feasibility of the proposal.
Okay, and then we've had a few questions asking about positive selection and what the positive selection criteria are, as well as how the process works. So I'll leave that one to Haley.
Yeah, so the criteria are the same criteria that we use for the full review. So if you can review that, it's in the PA. So, you know, what's the overall value of the project, rationale, the plan itself for the proposed award, if it is awarded, feasibility, and diversity, equity, and inclusion aspects. Yeah, I'm sorry, there was a second part to that question. What was it again?
The criteria and then process.
Process, yeah. So all of the applications get sent to the panel, as I mentioned, and they can essentially select out of that pool. So that's why the positive selection preview page is really important part of the application, because that is what they see first when they look through the applications.
And just to add a little bit, Haley had mentioned when she described this process that the scientific members of the grants working group, but also the patient advocate and nurse members of CIRM's governing board are allowed to participate in the positive selection process. So when you write your preview page, your positive selection preview page, and you're articulating the strengths and the value proposition of your research, you should think about how, what other ways it might stand out other than scientific, scientific innovation or whatever you're choosing to highlight. Think about how might your approach offer advantages that a patient could appreciate, or a nurse, or what are the DEI elements that might help it stand out. You have an opportunity to think of all the different ways to articulate how the value of your proposal in sort of a competition against all of the others. So you take advantage of that page to do that.
Okay, and then we have another question asking if some.
1 second. With the review criteria, did we mention feasibility? Okay, good.
Yeah. Next question is, can one pursue a therapeutic candidate and a biomarker related to it in the same application?
No, I think for every application, you have to choose one track. So each application is either a therapeutic track or a biomarker track. And each PI for a cycle can only submit one application. Within the same company, if you have related programs for biomarker discovery and a therapeutic discovery, you could have separate applications coming in, but you need to nominate different PIs to lead the efforts for that application. I don't know if you want to add anything to that.
I guess maybe we could add that it's possible that if you were developing a therapeutic candidate, some of your studies might lead to the discovery of a biomarker as part of that project. However, you will have chosen the therapeutic track and you will be evaluated on whether or not you meet the therapeutic outcome. And if it appears that you're too distracted developing other things along the way, that might hurt the feasibility. But if it's natural and it's part of the program and makes sense, it's not inconceivable that other types of discoveries or outcomes or tools could come along as part of the process of identifying that therapeutic candidate.
Okay, so the next question is, for a for-profit application, are we required to reveal the names of targets and compounds and other patent-sensitive information, such as chemical structures? Kelly, do you want to take that one? You're nodding.
Well, I was going to say that Haley could answer, but I can answer it too. So Haley can tell you more about the confidentiality of the review process. And of course, nobody can force you to reveal the names of your targets. However, if you choose to hide them, the reviewers might have a hard time evaluating the feasibility of the proposal if they can't look up or learn more information about a pathway or something along those lines. So it's a risk to not share enough information.
Yeah, just to echo Kelly, I think the short answer to that question is yes. I understand there could be some hesitancy from companies to reveal information, but all of our applications are considered confidential. All of our reviewers are required to complete conflict of interests. If you are concerned about exposure to other competing companies in some way, keep in mind that the application allows up to 3 exclusions. What that means is you can name up to 3 individuals or 3 for-profit organizations that whoever, you can name any one that you like, I suppose, or any company that you like. Some people will often put companies that they know are developing similar technologies or things like that. And we will honor that on the review side. So any person that has any affiliation, financial affiliation with those entities, or is this, person that is named that on the exclusions, will not review your application.
Okay, so the next question is, if a grant is awarded to a university PI, can this award be transitioned to a small business to continue the work during the lifetime of the grant? So I think, April, would you like to answer that one?
I'm going to actually defer to Kelly on that one, just historically, if there's been a precedent for that.
It is possible for the award to be transferred to a different institution. This is the precedents for it have been when a PI has moved to a different institution. It still has to be in California. So if an academic PI were to relocate to a company and wanted to take the CIRM award, you'd have to make a pretty compelling argument for that, and the indirect costs that were a part of that award would likely have to be cut because for-profits aren't allowed to have indirect costs on the award. So it would be something that would be submitted as a request and reviewed by CIRM to make sure it's feasible, to make sure that the new home of the grant has the right environment, and there's no reason to think that the work couldn't be completed there.
One other thing I should just note is that whoever is holding the grant must also hold the IP. So anything that's developed out of that may be relevant for profits.
Okay, and then we have another positive selection question asking who composes the scientific group that is doing selection out of that pool of applications, and how is it influenced by the expertise of who happens to be on the committee in that cycle? So I think this one really,
I can take that. So we formulate that group based on the applicant pool. So it is, it's based on sort of the overall, out of the hundreds of applications that we get, we compose the panel based on expertise based on that. So that's basically how the panel is sort of formulated based on different disease areas, based on different approaches, things like that. So those are all factors in terms of how we compose the grants working group. If you're curious about who is currently appointed to our grants working group, it's also available on our website. I don't have the link handy, but I think if you just search our website, you can also take a look there and see out of that. I mean, obviously there's a few hundred names on that list and they won't all be on any given panel, but you can get an idea of the kind of folks that may be on any given discovery or any of our panels.
And Haley, you might want to talk about the 15 grants working group that by law, and then we use specialists.
Yes, that's as well. So that doesn't necessarily bear on positive selection though. So positive selection is done only by the 15 grants working group members and up to 7 of the patient advocate and nurse members of our board. But definitely in terms of the full scientific review, if there is any expertise that isn't covered by the 15 panelists, we do bring in ad hoc specialist reviewers for specific areas of expertise that will also review CIRM applications as well. But that's for the individual full review at the second stage.
I didn't realize we were talking about the positive selection. So yeah, that's good.
yeah. So we also have another question asking a few of this one came up a few times. Will there be another grant cycle this year for DISC2? And then as well as inquiries about when the DISC0 deadline will be.
I can answer that one. So this year we will not have another DISC2. But as I mentioned earlier on, as you know, the board at the last board meeting, we at CIRM are undergoing a strategic allocation framework exercise prioritization, so that there might be changes in what we prioritize. So if we prioritize these two, there might be more calls. But this is to be seen as of September. We hope to have some recommendations to the board. And the next DISC2 is Kelly Chan, November, right? In November, we'll have application deadline for DISC2. And there will be a Remind X, which is focused on neuropsychiatric neurodevelopmental disorders, which is going to be in January, right, Chan?
We haven't announced that, but that's a
We are
good.
thinking around that, but
Yeah.
it's early 25.
There is a webpage for cirm.ca.gov/remind if you want to find out more about the Remind program in general.
And then we're hitting time, but I think, do you want to answer a few more questions or? Okay, one is that's come up a few times is whether DISC2 funds can be used for GMP production as an allowable cost.
Generally not. DISC2 is at a stage where you haven't yet really shown proof of concept for disease modifying activity for your candidate. That's expected by the end of the award, and it doesn't really make sense to invest in an extremely expensive activity like GMP manufacture for something that hasn't proved its merit yet. So generally speaking, no. There may be exceptions every once in a while. We've heard of situations where the GMP production for some reason is the same price as a research grade production, in which case, why not? But typically, no.
Okay, and then one last one is for previously submitted applications that did not get funded. Do they have to be submitted as a resubmission, or can it be submitted as new with more preliminary data?
Well, I guess if we're talking about a resubmission that received a score and critiques, I suppose you could choose to ignore that and put it in as a new application. But you'd be missing the opportunity to address the critiques that you received previously and have reviewers look at that. If you came in as a new application and ignored those critiques, it's possible that somebody on the review panel might have remembered it from before. It's not clear. I think it's usually to your advantage to try to address critiques if you receive them. However, it's up to you if you do that. If you did not make it through positive selection and you don't have a score to address, then it's just viewed as a new application regardless. It's not a resubmission.
Okay, so we're a few minutes over time, so we'll call it the webinar here. We'll be answering the unanswered questions will be incorporated into our Q&A, and you can also reach out to us at discovery@cirm.ca.gov with your questions, and we can respond to you by email. So thank you.
Thank you.
Thank you.